Welcome. Today we're going to talk about comparative modeling, and many of the slides come from Andrea Schafferhans. I rolled two talks into one lecture, so it's going to be a little bit of a fast forward. In principle, we know since Epstein and Anfinsen in 1961, that sequence uniquely determines structure. What it means is I take a sequence, I throw it into a solvent, it adopts a unique three-dimensional structure, and it does it always in the same way. If that is true, then in principle, I should be able to predict by first principles protein folding. Because the forces that make a protein fold essentially are in the connections between the residues, between the atoms. And by looking at the pair potentials, at all the potentials of what is happening, doing a molecular dynamics simulations, I could simulate this model in a way in which I sort of go downhill in an energy landscape, right? In principle, I should be able to do that.
Is that a huge assumption that only the sequence gives us the 3D?
I do have to put in another quote. It comes later. I made a mistake here, but there is a paper from '61 that showed it for some particular protein that this is the case. We know many proteins for which this assumption is clearly experimentally proven. So it's in that sense not an assumption. The assumption is that it is true for all the proteins. That in fact is also not the assumption because we know it's not true for all proteins. But the assumption is that it is true for many proteins. So maybe we cannot fold in the computer all proteins, but at least the ones for which we experimentally can show that it's true, and the ones that are similar to those, in some sense, in terms of not sequence similar, but in terms of the way they behave, maybe the short ones, this should be true. And in fact, the problem has been solved. The protein, what typically is referred to as the protein folding problem or the 3D structure prediction problem. The first solution was in the 60s, was reported in the Washington Post. The second was in the 70s, reported in New York Times, and then in the 90s again it was Washington Post. Okay, if you see, well, one thing is it is not very often that you get into the New York Times or the Washington Post with science. It's an exceptional reality. So this is really a big step, that's what you see. And if you see the same problem solved in 4 decades, you see that something is not quite right. And that brings us to this issue essentially, how can I assess whether I'm doing it right or wrong? So why do you believe this happened? Or how would you estimate, how would you assess whether you can actually run an energy simulation, molecular dynamics kind of simulation, and really correctly predict the three-dimensional structure?
If you're really certain that of the actual structure of a protein, like there are several crystal structures and NMR structures and everything, and then you give the computer only the sequence and some models on the forces, and it gives you the same structure as has been confirmed in experiments, then there's a good chance that it's correct.
Franziska, that is exactly the algorithm that these methods used. In fact, so I don't know the people behind the first two, but I know the group behind the last solution here. This is exactly what they did. So in that particular case, that was still in the 90s, that was still before we had several structures for one protein, but they used many structures. So they did not only do it for one protein. In fact, that article based on a prediction for about 50 proteins. And they had structures for those. So what you describe as a safeguard is what these people actually used, and still they failed. Because, in fact, they sort of optimized something in their parameters that was taken from the knowledge of those structures that they then looked up and actually found. So how can you avoid that? And in all of these cases, this is not a social event. It's not somebody who tried to cheat. It's not somebody who tried to get a very good scientific reputation out of it. They all somehow fell victim of internal over-optimization, and they had not realized that they do this. I can show you later maybe an example for how this goes wrong in a different lecture, not today. But the question is, how can we do it? So this algorithm that Franziska proposed is what they used, and it failed. It makes a lot of sense, but it failed. So how can we do it? Do you need an additional step? Anybody has an idea? And actually, this is the kind of go forward, and this is the kind of thing that is trivial, because I believe I already gave you the solution in a different context or in a different story. Anyway, most likely many people here in the room will somehow know something about that solution. Anybody? Let me just try to. It's not charging. Oh, okay. Yeah.
I'm not really sure if you want to have the fact that you probably need several structures. So
they had 50.
Or that there's this, and that you shouldn't know what your correct solution is, because there's
you shouldn't know the correct solution.
Competition
CASP.
Where you don't know that.
That's exactly the answer.
Really, if you use the knowledge of what the structure looks like for making an experiment, and say that's not really what I said. It was like you do it blindly and just sort of not use that knowledge and really only use the sequence. And if it doesn't give, if you have to squeeze the parameters so that gets right.
But the idea is, but you could still fix the parameters of the structures that are known today, and you try to predict for something that is not known today, and you try to predict for something that is not known today that is different from anything you see today. And this is in fact exactly what CASP, Critical Assessment of Structure Prediction, does. And CASP was put forward as an answer to this problem that I showed on the previous slide, so that people claimed or believed, not claimed, they claimed and believed that they had solved the problem. The way it goes is that the organizers have a period in which they talk to colleagues, and at some point I was a CASP organizer, and I did this. So you find whoever is an experimentalist, or you contact as many people as you can. You ask them, over the course of the next few months, are you going to have a new structure somewhere in your pipeline that you today actually do not know yet, but you can estimate that within the next 3 months or 4 months you will know the structure. Are there any cases? If so, and that's what the organizers collect in the phase between April and May. And then in between June and August is the prediction phase. So all of these collected cases are given to people who develop prediction methods, and they predict. The deadline for submitting your prediction is the moment that the structure gets to the level of a manuscript. So the moment that the experimentally determining group, it leaves the experimentally determining group by them submitting a manuscript, okay? Then somebody else in the world will know the structure, and that's the deadline for the predictions. Then there is a period between September and roughly November or something like that, in which a group of expert assessors look at predictions and the solutions, and actually more solutions are also collected. Not all structures are known in August. Some are collected over the course of September, October, November. And then there is a meeting in December when we all discuss with the groups who predicted, and sometimes with some people who did experiments, how well it does. So one success of CASP is, and this is the first 10 years of CASP, is a biannual meeting every 2 years. In the first 10 years, you see the number of groups participating here in blue is rising continuously. It sort of plateaued at some point, but CASP is still a very, very actively evolving community, actively evolving process. What became clear is that only homology modeling or comparative modeling, and again this will be the theme of today, really is working in the sense of getting a good 3D structure prediction. There is no general prediction from sequence yet. MD essentially does not work on that level. And there are many, many, many important improvements over the years in many aspects of the structure prediction. This issue here about we cannot do MD, I will possibly at the end of the lecture, maybe this time the lecture is too short, get back to attempts of doing this with general purpose or special purpose machines. There's a particular machine in New York called Anton, and Anton is an approach from David Shaw and colleagues to build a parallel machine that is specialized in terms of hardware, software, and everything to solve this problem of fast MD simulation. And at this point, Anton is many times faster than anything else that exists in the world. And so they can do it further than anybody else, and they do quite remarkable stuff with it. Now, one type of improvement, one type of conclusion from CASP was that it really helps to connect servers. So the more servers, meta servers, the more you connect, the better. there are problems with CASP. One of the problems is that the number of samples we collect is limited, so typically it's in the ballpark of 100 proteins. So here's another problem illustrated. Same method, A uses 53 of these 100 proteins and gets a performance of X. And now you have a method B that uses 35 of these 100 proteins and gets a performance X plus a little bit. And now the question, so Y is a positive number here, is B better? How do you answer that question? But let's begin first. For the first part here is 100 proteins. Is that enough?
It depends on what's your certainty.
That depends essentially on something like the standard error. That depends, and that brings us back to the issue: how big does Y have to be? So method B is X plus Y. So how big does the Y have to be? If it is within a standard error, then that is not big enough, right? So we have to clearly have a standard error estimate, and that's one important issue here. And 100 proteins is not a lot. But the next issue here is another complication, namely that we compare apples and oranges. So this is a subset, the 53 are a subset of 100, and the 35 are a subset of the 100. They may have zero overlap, right? So the comparing two completely different data sets is unclear how to handle that. So these are major issues, and this brings us to one question. Say we had one particular CASP, and in this particular CASP we had 7 different methods, and these 7 different methods chose 20 different, at random from the 100 they choose 29. Okay? And now when I compare these 7 methods, for some of the 29 they may be similar, so some of them may predict exactly the same protein, but some of them are different because the 29 are chosen from 100 randomly, okay? And in the second experiment I only compare 11 identical. So 29 is a much larger number than 11. So I'm trying to compare. Let's go to the second extreme here. I have 11 proteins. And you know that there are about 120,000 structures known. And now I'm going to compare 2 structure prediction methods based on 11. So this is a tiny number. Okay, 29 is also not such a huge number, but it's almost 3 times bigger, right? So which one of those is better?
Well, can't say just by the numbers because the 11 they are identical. So if we are trying to predict,
oh yes, you can.
This type of,
so this answer here is that it does not need any further assumption. There's a very clear answer for whatever we tried. So the answer is clear in this particular case. In this particular case, the answer is one of these 2 is better than the other. In fact, much better. And the question is which?
Well, if I'm looking for a specific protein that corresponds, that's identical to those 11, I'll choose.
No, that's not what I'm looking for. So my criterion for which of these 2 is right is going to be 500 different proteins. Because I actually, for the same 7 methods, and I'm not going to show you the results, the next slide has the results, but the background underneath here, this is secondary structure prediction. And for these 7 methods, I in fact had 500 different proteins the prediction. So from 500 proteins, I knew really what method was best. Okay, I only asked for these 100 that were in fact at one particular CASP, I can't remember which CASP, 4 or something like that, at one particular CASP, those were exactly the targets. And these numbers were chosen because roughly this was the kind of overlap by which methods were. So on average in that CASP, methods picked about 29 of 100 targets to work on. That's how that number was chosen. So which one is better? 29 or 11?
11. I'd say the 11.
So the small number. Is anybody agreeing with Anton? Small number. Hit him.
Yeah, but they're identical.
They're identical. It's amazing. It was way, way, way better. So both of those were obviously 2 small numbers, but given the small number, this gave a much closer answer to what we observed over 500, and that's the real data. Another way in which you can do that better, and that is in fact what what we learned at CASP from that experiment is when you compare 4 different methods, you're not comparing on one data set. What you're going to do is you're going to say, I look at the subset of proteins that A and B have in common. So for 7 methods, it may be as small as 11. But if I made pairwise comparisons, maybe choosing randomly 29, for many of these comparisons, the overlap will be more than 11. So it will be at least 11, but for many it will be larger, right? So that means that for every comparison here, essentially I ask, is method A better than B, and is that better significant? And the degree of better is the darker blue, the better it is, right? And then simply now comes the next issue. How do we get from here to a rank? Well, one way of doing a rank is simply you say the method is number 1 that is better than most, so we count for every single method, how many is it better? It is better than, in this particular case, 1, 2, right? B is not better than any, C is not better than any, in this particular case, D is also better than 2. So now we would have to get into seeing that this is darker blue, this is lighter blue, so D is slightly better. But that clashes with this point here. This clear white means you cannot distinguish between A and D. So essentially, in that particular case, it would imply that A and D are rank 1, right? That's one way of doing it. And there are other ways in which you can do that. But the important thing is that the take-home message here is you have to really compare things on the same set. And for the same set, you have to, and for CASP it's clear, because the proteins were not known before, if you assess methods that have been published in the past, you have to make sure that you compare them on the testing set, not on the training set, not on data sets that are optimized. Okay, how do we get to 3D coordinates? By experiment. So this is sort of detail, this is the simplified version of it. We, at this point, have 120,000 structures in the PDB. Protein Data Bank is the data bank, as you all know, of high-resolved structures. Of these 120,000, about 109,000, 110,000, so the vast majority are done by X-ray crystallography. The way you do X-ray crystallography, you essentially get the protein out of the cell, you get it in a very high concentration. The concentration is so high that actually the protein grows a crystal. A crystal means you form a lattice here of many protein molecules, right? So the order in the crystal, in a carbon, in a diamond crystal, essentially you have a carbon at every single junction. Now here in the crystal you have at every point a protein. Through that you shoot X-ray beams, and they have to be highly focused X-ray beams. So that means this is for large X-ray sources such as CERN. In fact, now what used to be used mostly for high energy physics, these big beams are beginning to be mostly used, or some of them are mostly used now for biology for exactly this sort of thing. Yes?
Normally the resolution is about like a few angstrom.
Yes.
How do they get the resolution with the X-ray? Because it has to be kind of in the same area of the wavelength. So how do they do that?
This is in fact exactly it. That's exactly what they use. The wavelength is not angstrom. So again, actually, this is true what you say. But you have first of all you do a phase transition. That in fact the X-ray phase length is below an angstrom. And I'm going to show you essentially. But what you get from so you do a phase transition, then you get essentially an electron density map. And this is I'm going to show you later an example for the resolution, and I'm going to show you an example for an electron density map in a little bit more detail. Hopefully you can see it with the slide. But essentially, you get an electron density map. Now from the electron density map, that's essentially the only real data you have about the structure. And now you have to fit the atoms into the electron density map. I'm going to show you an example for that. Through this fitting, you actually get an atomic model. Now, the other major method out there, so one issue with crystallography is that most proteins, although the cell is very dense, they are not sitting in the cell as crystals. Okay? So getting to the crystal requires a lot of manipulation, let's call it that way. NMR has the advantage, nuclear magnetic resonance spectroscopy, has the advantage that you can essentially put a protein into a tube, in a pipette, in a tube, in the same solution, essentially, in which it sits in a cell. Okay? So here you're not growing a crystal. You're essentially taking a protein out of a cell and put it in a magnet, into a magnet. Now, the problem of NMR is essentially these magnets are big, and again, now they're even bigger. A few years ago, a 900 megahertz machine was the biggest thing. Benjamin, you're right. Just for scale, so here you see a person up here. This whole thing is the magnet. This in City College or in the New York Structural Biology Center, this entire room here is shielded, full of NMR machines. You walk into this place, and anything that you carry that has some electronics in it, like your credit card, is wiped. And this, to give you an idea, this is about the same size as the downstairs hall. So it's a huge space. And this entire huge space is wipeout space. And it happens all the time, by the way, there. But people sort of get the privilege of doing new credit cards. Anyway, what you get out from it is you essentially see distances between protons. And these distances between protons, ultimately what you see is a particular arginine, is it next to an alanine? Now, when you know that an arginine is next to an alanine, you know that every single protein has more than one arginine, typically, and more than one alanine. To resolve which one that is an additional experiment. You try to label them, you try to sort of shake them a little bit, to do something in order to distinguish between this arginine and that arginine. And that is a lot of additional problem that goes into it. And then, at the end, you still only get what an NMR spectroscopist call models, and in this particular case you would have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, and so forth. You can count them yourself. You see that some regions, all the models essentially converge to the same solution. Here in these regions, and in some regions the solution is more different between the different models. NMR spectroscopists argue that the regions where they converge all to the same, they are more rigid, and the regions where they are not the same are more flexible. So that essentially what you see by the differences in these models is functional movement. Okay? And again, at this point there are about 10,000. When you come to the point of having a good crystal, a good crystal that affects very well, in fact that is the major criterion for the resolution you get with these X-ray beams. When you have a good crystal, from that moment to getting the real 3D structure is essentially largely automated. There's still a lot of fitting. There's still experts looking at how to fit into the electron density map. They optimize using all kinds of prediction methods. They optimize using programs on the screen. There's something to be done, but typically this is ballpark maybe weeks. This one here, going from the raw data to the structure, is at least 3 months for absolute experts. So this is not something that, just put it down, on the level of a PhD student is easily doable. Still not. So this still requires more experience than that. So postdoc and professors take 3 months on getting from the experimental data to the structure. Typically, that's the ballpark, right? If we have a new structure, not if we reproduce something that already exists, where you simply see, is my data corresponding or similar, does my data fit to what I already know, then things are faster. But if you do a completely new structure, this is still several months. In a good case, okay? In bad cases, it is way more complicated. Now, one level down in resolution, cryo-EM, electron microscopy, cryo, so you freeze it. This number is most likely the one that is rising the fastest of all of these. So the future will have more cryo-EM images. But what you see at different levels of resolution, so at 4 angstrom, 8, 16, and 32, essentially what you see is a very coarse-grained image of the surface. So EM only sees the surface. In some cases, you see that even here at the 32 angstrom, you still see a hole. You somehow see inside the white here. At a higher resolution, you see actually that there's something in the hole, but you can get through. At the highest resolution, you see balls. or spheres, or not really spheres. You see the outside of the sphere, right? So at this point, you see something about the surface, you see some of the structure, but you don't have an atomic model. So this is not what you get, but this may be functionally irrelevant already, and you may actually be able to couple this with computation. So you may be able to say, well, for some of those structures underneath I can do comparative modeling. For some of the structures I can sort of do some energy minimization techniques. And I have constraints. I know how the surface must look, so I know what the MD simulation has to end up with on the surface at least. So I could possibly hope that the ones that are compatible with this surface are only possibly short bits, and that they are only the only ones that are compatible are the right solutions. This is what people do. This slide here is from from Chu, but the major methods now come from the Charlie lab. I will get back to the Charlie lab when we talk about comparative modeling. Here, this image of electron density that I talked about. I'm not sure how much you can see at this point. The blue is the electron density map. We are talking about a ring in 3 different resolutions: 1 angstrom, 2 angstrom, 2.7, and 3. And let's look at the extreme example here. At the 1 angstrom resolution, you may see that the ring, you have even electron density here in the ring. There's black inside. So this ring there's no other orientation of the ring that is compatible with the electron density. At 3 angstrom, and now this is the point where we would have to have a darker room, essentially what you see is it's sort of a rod-like mesh here. I'm not sure how much you see in the back. Yeah, you can't see it at all. So imagine, this here is imagine a metal mesh here in black, and the metal mesh is essentially giving you a rod. Now, the way the ring is oriented, whether you're oriented this way, even the movement in this direction, is all completely compatible with the mesh. So here you have many different orientations of the ring that are all compatible with a 3 angstrom resolution. So the structure that you then find in the PDB is a model, because somebody said this fits best, or it fits best because of the other constraints about the next side chain that clashes with it, or because of you don't want to stretch bonds too much. But here you still have movement, here you still have a mesh, and even in the case of 2 angstrom, you have a mesh. So in this particular case, the mesh is now no longer a rod, but it is a sphere, meaning that you can still turn the ring. But in this case, I believe there you see the blue, right? Or not? Do you guys see anything? Oh yeah, there you see the blue. No, you don't see that there's black in there? So in this particular case, for 1 angstrom resolution, you really have a very well-defined structure. There are several structures in the PDB at that level of resolution now. It's still in the hundreds of the 120,000, but still, so increasingly we sort of reach this level. Again, 3D, 2D is context. That is what NMR essentially gets to get 3D structures. And there's 1D, for instance, secondary structure. Secondary structure is stabilized by hydrogen bonds. And that idea, that very same idea that hydrogen bonds stabilize secondary structure and that these lead to 2 different types of secondary structure. I made a mistake in the quotation here. So that one is helix and one is strand is in fact what Linus Pauling published in a series of papers in 1951 and 53, and that got him the Nobel Prize in 54. And again, so the idea is you have the helix here running around. There is a hydrogen bond formation between the backbone of residue I and I plus 4, I plus 1 and I plus 5, and so forth, right? Move it through. For the strand, so this is 2 strands together forming a sheet, 2 fingers forming a sheet. And again, every other residue I plus 1, I plus 2, hydrogen bonds to another pair somewhere else, okay? And that could be J and J plus 1, J plus 2, or minus 2, depending on the orientation of the other strand. That much is clear. In the cartoon you see these as the sheets here, and another representation you see them as the arrows and the rods. When was the first structure determined? Protein structure.
Is it insulin?
No, it was the third. But it's amazing. Who asked that?
Hemoglobin and
Who asked that? Hemoglobin and myoglobin. Who said that about insulin? It was the third. Amazing that you know that. No, but it was not the first. It was hemoglobin and myoglobin. But when? Again, Pauling's Nobel Prize for predicting helices and beta strands was in 54. When was the first structure determined? You may argue that it must have been before that, because they cannot give out a Nobel Prize for something that hasn't been observed yet. But in this particular case, it's not true. In fact, myoglobin was the first 58, Perutz, the hemoglobin. But Perutz had started the technology. Kendro just had a better ability to handle myoglobin. But essentially, it's the same lab. Very similar structures. This is a dimer here, homodimer. Does anything hit you looking at these structures?
Almost only helical.
All helical. So the first 54 was the Nobel Prize for essentially seeing beta strands and alpha helices, and now we have 4 to 6 years later the first structures, and they don't see beta strands. And then comes insulin, which is another 7 years later, after this, so 67 roundabout, and it's all alpha helical too. So that was the moment when people felt, okay, he got a Nobel Prize for, he was right on one, he was wrong on the other. Now we know both exist, so Linus Pauling was right. How do we annotate secondary structure from 3D coordinates? How can we do that? So I give you 3D coordinates. How can you get secondary structure out?
We should be. We should be able.
That is absolutely true. So I'm not, you already know that we're going to talk about secondary structure, but you anyway know that there are secondary structure prediction methods. So actually, no, that is not a giveaway. But yes, we are able. But how?
Calculate the distances between atoms, because actually in the X-ray we don't have the protons, so the hydrogen atoms. We know oxygen and nitrogen.
So wait a minute, X-ray we have the hydrogens.
Do we have?
Yeah, we have the whole thing.
Okay, I thought we inferred it from the other. We calculate their distances and we know more or less the length of the hydrogen bonds. So when they are in the correct distance one from another.
So historically, it's an interesting way that you saw that, but historically the first idea really was more in this. So actually the first secondary structure prediction methods were done on experts looking at it and guessing the secondary structure. And that was sort of more aligned with the idea of using geometry. So something that looks like a helix, you call a helix, right? This is your solution is in fact what is mostly used, the program called DSSP, that essentially looks exactly at the hydrogen bond. And then it establishes sort of different rules for hydrogen bonds. So you see the pattern I and I plus 4, I and I plus 3, I and I plus 4, 5. You call that 310 pi or regular alpha helix. And you see something like beta sheets in the right way. The hydrogen bond is defined as less, so you have they're closer than something, and that defines the energy of a hydrogen bond. And if it is less than whatever threshold, then you call it a hydrogen bond. That's exactly the idea. So it's following Linus Pauling. There are other methods that existed at some point here. That's from Dimitri Frischmann, who is also in Munich, as you know, at the TUM STRIDE. And essentially the idea there was, let's take expert assignments and do a machine learning in expert assignments. So let's just see what experts learn, and let's assign secondary structure according to that. And in mass is the idea, let's not design secondary structure based on the 3D structure. Yeah.
I just wanted to ask why you don't use the angles, because of course thought that also the Raman-Hunter plot has...
There's a method that you essentially define No, but yeah, define used at some point used angles too, and there's another method that uses angles too. So this has also been used. But ultimately, so I believe why DSSP became ultimately the winner is because the principle that makes the formation of hydrogen bonds it makes the formation of secondary structures hydrogen bonds. So assign hydrogen bonds according to what makes them form ultimately is in some sense the most reasonable way of doing it. And I believe this is why DSSP got through in the end. There is this issue here of continuous secondary structure assignment, and I believe we're going to skip that too. In one analysis from Natalie Kollock, they looked at different assignment methods. DSSP defined a P-curve. P-curve, in fact, is exactly the one that is based on angles. And said that overall, in the three-state system, helix E is extended, strand, and L is other, or loop. They agree to 30% of the cases only. So there is quite a difference between these methods. And again, when Natalie published this 2 decades ago, it was quite not clear what would be the best. I believe today everybody accepts that the solution is DSSP, yes?
So there was a 30-year gap between the first structure and the first secondary structure assignment method. Because that was 83, right?
Yes, because in between, exactly. So we have to be, so DSSP is the, no, I believe I skipped DSSP all.
That's 88.
There's not even the name written. So DSSP is the dictionary for secondary structure of proteins. And what they mean by dictionary is along with the method, they actually for the entire PDB published an appendix and had a secondary structure assignment. So they applied their method to all the proteins in the PDB that were known, and I cannot quite remember what the number was, but the number was in certainly sub 100. My impression is it's sort of 83 was in the ballpark of 20, 26, so something in that ballpark. So yes, it took a long time to get 20, 30 structures. That's the problem. So in 94, I remember we had 126 structures. That's one number that I happen to remember. But the beginning, the first years really moved very slowly. And that ultimately is one answer. The other answer is that crystallographers simply believe that they can anyway do it better. The third one is people did not really apply a lot of computers in those days for issues such as this one. So if you see it on the screen and you can do it, you don't need a computer. So I believe DSSP is in some sense arguably also a change in culture, arguing that, and this was also very much intended from Wolfgang Kapsch and Chris Sandor, arguing that you need, a computer is able to do certain things more objectively than a human. That's one point they put down here with the publication of DSSP. And I believe today we would all tend to agree, but that was a battle for over 10 years. No, more than 10, much more than 10 years that people did not agree with that notion, that a computer could be better, more objective than a human. Okay, let's get to comparative modeling. Let's get to sequence to structure. That's the image that I missed before. Christian Anfinsen, who got the Nobel Prize in Chemistry in '72 for work that he started in the 50s, and the breakthrough really was showing in the early 60s, that just the sequence leads to 3D structure. If so, you would expect that similar sequences somehow have similar 3D structures. Let me show you a couple of examples here. KRAS, in fact, historically, the first, it's an oncogene, is the first protein that was involved or was known, or first gene that was known to be involved with cancer. This one is in human. This is the way it looks. What I show here, and these slides come from Andreas Schafferhans again, what I show here is the non-native version, where for residue 12, position 12, you change the glycine to a cysteine, the G to C. Okay? That mutation causes cancer. So that's essentially the mutation that leads this oncogene to become an oncogene. And you see that the two, or you may see that the two, the overlay, that for most places you do not even see a difference between them. You do not even see two. Here, in this loop here, you see two. You see a blue and a red. But throughout, these two look very similar. You do also possibly appreciate that the binding site is slightly different. The ligand is bound differently, and that, in fact, is the reason. It's subtle. But that was enough here to sort of increase the odds of doing something that is not quite right and that ultimately leads to cancer. Now, let's look at a homologue of it, the protein RAS, in Lyme here. At 85% sequence identity is a protein in human. And again, RAS, and this is the native version of RAS. Again, in the previous slide you saw there's no difference. But now you see the helices here go apart, but then you see other places here. These two helices are still very similar. The beta strands are still very similar, right? You see the binding site now moves more, the loops here move more, but there's still a lot of similarity. At 85% sequence identity. Now we go down further. We go to fly. We go to 28% sequence identity. And again, essentially these sheets look the same. These helices begin to move. Helices move here more away than the other two. The binding site looks very different, but still very, very similar in structure. We move one level down. We go down one level down, even more down. We go to 19%. We go to bacterium. Now we go to something that no longer has anywhere near the same function in some sense, but it essentially still, so it has a binding site, but it does something, it binds something different, has a different role in the system, but still, the scaffold of the structure at 90% is still very similar, right? Now, in terms of the HSSP curve that I showed you, we are sitting somewhere below here, where from sequence alone, we could actually not infer that these two have similar structures. Now, comparative modeling will try to use sequence similarity to predict 3D structure. How can that help us? So one way in which it can help us, here's the method conserve, that essentially all it does, I don't know whether you have seen it ever, you take a protein structure, you take an alignment of a family, and you simply plot upon the structure, on the surface of the structure, the regions that are conserved in the alignment. So more conserved is darker, dark red. The least conserved is dark blue up here. And what you also may be able to see here is a binding of substrate, and that the binding site, so the yellow binds exactly where the darkest red is. Yes?
And so you've showed two superimposed structures and superimposed structures that are a little bit off. Those are soluble proteins, which means they move in solution. And my question is, the difference in superposition, is that actually meaningful compared to how the protein actually behaves in solution? It moves, right? So this is a crystal, right? This comes from.
So the question, I cannot answer this question. Okay. So one way to answer this question is maybe in the purple one here, the binding site that I had before is no longer possible because there's a clash of the loops, because the molecule no longer fits into it. Okay, then you would predict that in fact for the purple one, it does not bind the same thing. Now you observe that the purple one doesn't bind the same thing anymore. Okay, you know that the functional movement is making this loop move a lot. So now back to your question. Does it make sense? Did I learn something from the move? In this particular example, the way I constructed, the answer is clearly yes. I did. How can I explain that? Well, I could argue they all move maybe in a similar way, and they all move around a sort of starting point, and what you see here is the starting point.
Also, would like to add what mostly moves in NMR structures are the loops, and this is mostly helix and beta sheet, and they tend to be more rigid. Even if
yes, the complication is
so basically
how rigid.
The complication is that we're getting into problems of averages here. What is most flexible in every single protein is the loop. What is most rigid is a loop in a binding site. And there we get into the first clash of words, right? So the other thing is that loops are somehow defined as the thing that is least consistent, which builds in this idea moves most, right? So now we get into a vicious circle of the definition. We define loops, so is this, well, here I may not, here's an example. Will the way it comes out of the helix, is that still a turn of a helix or is that a loop? And typically the way to decide that is because of consistency. And again, you see that we're entering a vicious circle. We are defining the loop by it moves more. So this is not as easy as you say it. This is the way people often answer that, but it's more complex. Yes, Harry?
I don't know that what we actually see is mostly the backbone.
That's correct.
So if we actually...
Here you see only the backbone, yes?
Yeah, if we have the actual residue, so it has polarity, it has functional groups, some have sulfur or another. So it may actually change the chemical environment in the pocket.
Yes, that is true. Let me get to that in a moment. Are you following up on a question?
No,
so this actually, anyway, so what I'm trying to say here is you can maybe build a model in order to see the conserved surface, and that may already help you a lot. Here I'm going to show you something where we have a model, and so this is the structure, and this is the model. This is an archaeal protein, and this is a human protein. This is the same structure as that, it's a homology model. Okay, but you see that the features, the electrostatic features of the two molecules are completely different, and that ultimately goes exactly to your point. This is why are the features on the right completely overall positively charged, left is negatively charged? Why are they so different? Because of the side chains. It's the same sequence, it's the same backbone, the backbone overlaps. So in the plots that I showed you before, these two would actually look the same, but not on the level of the side chains, or not on the level of a method that actually considers the biophysical features of the side chain as electrostatic potential, as you see here in grass. And that's from Diane Murray and Barry Honig. So electrostatic potentials are typically plotted by GRASP or GRASS, and that comes from Barry Honig. So we can learn from models, and then another thing that we can learn is how structures are rearranged. We can learn from models where in the dictionary of all structures you sit. But how can we predict it? So what's the idea behind comparative modeling? Is it trivial for you guys? Is that something that you have heard hundreds of times? I believe I've said it in passing. Yes, is it all very clear to you? No?
If you mean homology modeling, then yes.
Yeah, so again, the lingo, we will get into that in a minute. So again, we have a query protein sequence, we have a protein in the PDB. This one here, for the protein PDB, know the structure and essentially infer that this here is the structure that you have. The reach of comparative modeling, and I'm going to fast forward here. So what I mean is the entire circle here is essentially all the known proteins. Then sort of that is an estimate, that is an overestimate, but let's just put down some fraction. So PDB has 120,000, UniProt now has 85 million. So there's some fraction that is experimental, okay? And then there's some fraction where we have a protein in the PDB that is similar to my protein but not identical, and by simply assuming that U and H, if they have similar sequences, they have similar structures, for instance by this threshold function, the HSP threshold, essentially as long as you're somewhere in this zone here, you can do comparative modeling. Essentially homology modeling, you can take the structure that is experimentally known and sort of expand this view. By going a little bit further into the twilight zone through sequence profile or profile alignments, you can push it a little bit more. And then you come to a point where roughly for half, a little bit more than half of all the proteins, you can actually predict through comparative modeling some region, right? Then there's the midnight zone I talked about, that we will not reach, but we will reach sort of something that is more than half. The view is slightly different if I look at entire organisms, a little bit less than half is what this one says. But essentially, for about half of the proteins we can do that, and the good news is, so this fraction is increasing over the years. Again, the lingo, comparative modeling, homology modeling is sort of the same thing. Historically, and some people still use that as homology modeling. I talked about homology actually being defined on the level of organisms, or on the level of parts of organisms, so not really on the level of proteins, because for a protein we don't really know the entire tree. This induced some people to say, well, the word homology is misleading. Actually, what we really do is we compare two sequences and use this comparison to model. In some sense, this is the more modern name. This is sort of the more historical name. But in the literature, you find this still more often. The other part of the lingo here is the target is the protein in the query, the one that you try to model. The template is the model, the structure that you have, and you try to map the target to template or two templates through the sequence alignment. So first, you have to identify the template. How do you do that? By, well, you begin by a simple blast search, by pairwise kind of search, and then you improve, and then you align the target and the template. And, well, again, you can do a more detailed alignment here, sequence profile search, and then you build the model. Now, in the simplest way of building the model, how do you do that? What's the simplest way of building a model? So you have a protein for which you know the structure, experimental structure, and you have a protein for which you do not know the three-dimensional structure, and you have an alignment between the two. How do you build a model? In the simplest way.
You just assume that the aligned parts have exactly the same structure.
You copy the coordinates, exactly. So you have corresponding residues. And for these corresponding residues, you just copy the coordinates. Now, then you have to assess the model. How do you do that? So you look, for instance, at regions and see that there are clashes of bonds, that suddenly two positive charges in the model have to be next to each other. It cannot be true, right? So you see that bonds are too stretched or too extended, and then you try to refine. You try to refine if it's too quenched, you pull it apart. If it is too stretched, you pull it together. If there are two positive charges that are next to each other, you sort of move them a little bit apart and see whether that is doing better for the model. That's the kind of thing. And I'm going to show you a few more details in a moment. But just the overview, when we look at the sequence similarity or identity between the template and the target, so the known 3D structure and the query, when it's very, very high, the major problem is really the speed. It takes some time. When the sequence identity falls, the quality of the model becomes the problem. Falls further, the problem becomes the alignment accuracy. So if the corresponding residue that you copy the coordinates of is not right, because you make a mistake in the alignment, and that begins somewhere here, in this realm, you begin to make mistakes in the alignment. And below there, the problem really is to in fact detect the right template. So you no longer find. There is a structure in there that you could have used, and that would have worked, but you didn't find it. Okay? These are the kinds of issues. So you use PsyBlast, HHBlitz, and these kind of approaches. Then you align again through more detailed methods, including profile. And let's just look at two state-of-the-art methods. The first one from Andrei Shali, who has been very successful with this. So that's the package modeler interface of the website. The pipeline, you find the structure, you do the alignment, you build the model, you assess the model. If the model is okay, if the model is not okay, you try to change the alignment. Or, in the most extreme case, you even try to find another template. So maybe the protein that you found is most similar in sequence, but maybe there's another one that would actually fit better to you. Or if you have two positively charged residues next to each other, maybe you move the alignment of the corresponding residues and you align slightly differently. That's the idea behind it. So you have spatial constraints that you extract. For instance, the simplest version, you extract all the C alpha distances for the model, right? And you know that somehow underneath here is the distribution that you expect. And if this is not the distribution you find in your model, then the model is not right. So it could be that you to have two Gaussian peaks, but that they are too far apart, or that they are too close to each other. They do not fit the expected distribution shown in black here by the lines. And if that is not the case, you try to move things around. And you iterate over this many thousand times. Since you just have a limited number of atoms or of residues here, you can do that. And the things that you move around is just a few. You can relatively quickly do that many thousand times. You can improve over this simple way by doing molecular dynamics, Langevin dynamics, and all kinds of other simulations, which they in fact in Modeler do. They run optimizations, they explore different types of doing the alignment in the model, different local alignments. The type of errors they do is they get the incorrect sidechain packing, they essentially sort of get loop strong, they do misalignments, they use the wrong template, then they get really wrong. Here's a statement, or here's for 3 proteins for which they know the structure. They test whether the dope score, the internal score that says this model is the best. So you move things around so that they fit to the distribution, for instance, of the C alpha distances. And then when they fit, you compile a score of how good they fit, right? They use many other things. And that's in the accumulated score here, the dope score. When the dope score is lowest, you have the best fit according to your assessment of how good the model is. What they show on this axis here is this C alpha, so the residue, RMSD error. And ideally, if the thing would work, the lowest score in your dope score, in the y-axis, would be the lowest RMSD, right? So, which is the case here, right? In this particular case here, you see that their lowest score, there's something left of their lowest score, meaning that the one they pick here has more errors than the best they could have picked, which is this one. But in a real case, they would make this mistake because they would pick the one with the lowest score here. They would pick this one. But that's a tiny mistake. Here in this particular case, you see that in fact is the lowest score. That's the one they would pick. And that is in fact the best. So here in this particular case, we move from, 7 to 4. So in this particular case, we would make a larger mistake simply by sort of refining the model, right? Or simply by the words are complicated to use here. Simply maybe there would have been a way to measure the model. Maybe there would have been a score that would be better. Maybe not. So what if a loop is missing? So I have the template has this loop, and my query protein has a longer loop. How can I do that? So in this particular case, I cannot copy the coordinates because simply there is nothing aligned there. There is no coordinate. Yeah?
As far as I know, in this case, it's just done by sort of applying forces to it and just wiggle it around. And
that is one way, but the first way is not that one. So first what people try, so the first solution in fact is find all the similar loops. So you have these 2 anchor points, and then you see the loop in between. Do we have in the PDB in other structures loops of that length? And then how do they look? Right? And then they sort of find the optimal path through the set of loops that exist. And this is very fast and very easy to do. Okay? That's the first. Only in the case that really there is this type of loop is completely missing in the database. Doesn't exist, right? Only in that case we do exactly what you said, Franziska. So we do essentially a molecular dynamics simulation, which in this particular case we can hope to do because now instead of having a long protein of 100 residues, we have an 8 residue loop, right? So do molecular dynamics where we have 2 fixed points in the loop in between. That's a much simpler problem than the one of really folding an entire protein. And indeed, so this is the loop length from 0 to 14 residues. This is the RMSD mistake. So at some point here is difficult where we put our hands in it, where we put the threshold. I believe they published, they said 14 residues, so they argued that all of these are still okay. But I believe the plot ends because they said that more they cannot do, but they argue that until this point they can do it. So until, I believe, maybe 14 is a little bit of an overestimate here, but for 10 residue loops or for 6 residue loops, this is doable. For very long loops, this is no longer doable. And whether that graph now is completely reflecting reality or slightly over-optimistic, let's not discuss this. But for some case, for short loops, we can clearly do that. For very long loops, we cannot, and very long, maybe 10, maybe 20. Okay. Another package is Swiss model. That comes from Basel, from the Schwede lab, and that's Thorsten Schwede, who has an immense impact on computational biology. Mainly, so his history is actually he's an extract crystallographer, but he completely changed the world through Swiss model. Originally, Swiss model came from Manuel Peitsch, Nicolas Gouet. Thorsten joined the team later, and the original, that's the website, the original philosophy, in fact, from Manuel Peitsch had been, so first of all, it has to be fully automated. It should be for non-experts, so meaning typically experimental biologists who do not know structures or do not know computers. And Manuel also had this point, if you do less, you make fewer mistakes. Which is a particular way of looking at life. Essentially what it translated into is do a simple alignment. Those days, in fact, that was BLAST when they started. Copy the coordinates, so what we discussed before, and then call it a day and publish. So all of these optimizations that I showed you from the model of the software, they just didn't do. Instead of this optimization, there was some text that said there will be many errors, find them. It was phrased in a different way, and I'll get back to that theme in a moment. But ultimately, that was the original publication of Swiss model. Meanwhile, so this is a few years ago. Meanwhile, as you can already sort of see from the from the flow chart here, things got way more complicated. And I'm going to zip through the whole thing and just say that today Swiss model is still automated. It's much more complex. It's very, very, I believe in many ways it does much more than Modeler by now, because the Charlie group is focusing on some other aspects of using Modeler, and they really have done a lot of experience to refine the Swiss model experience. And again, it allows you to model at this point 40 million proteins, and they are available through the Swiss model data set online. There are other tools out there, and I believe I'm going to quickly go over some of those. The first one, HHPrET, that comes from André Lupas and Jonas Söding. That underneath of it is HHBlitz. I briefly talked about the major step, really, in fact, for the improvement of other modeling servers is their alignment method. The profile alignment, that was sort of a major part of making that better. And they do, in fact, do many things different, but then at the end of the day, you see run modeler. So ultimately, the major improvement that you get from HHPrET is do better in finding the right template and doing the alignment for the right template, but then the sort of energy stuff is done by Modeler, and with that they developed something that in many cases really did beat everything else out there. Another idea is 3D Jigsaw from Paul Bates and Michael Sternberg's lab, and there the idea was really chop the protein into parts, into little parts, and see how these parts would fit. So not do a homology model on the whole thing, but find pieces from other proteins that are looking like they fit into your proteins, piece it together by fragments. And once you have the fragment, you identify all the fragments, you assemble the fragments to a structure, and you get a homology model. That's the idea behind that. There is WhatIf from Gert Frendt that has a large community because it has a nice visual interface. Here are some of these pictures from Elmar Krieger. So they have there's Yazara underneath. One of the strength of this method really is the beauty of looking at the structures they produce. So you essentially could possibly run a modeler and use their interface. But WhatIf is more of a method to use to play around with protein structures to optimize while you go. So WhatIf is very, very far away from an automated tool. Modeler is much more automated than WhatIf. WhatIf is meant to, what if I change that residue? That in fact is the reason for the name. How would the structure change? What if my protein had a variant here? How would that change the binding site or something like that? And this is a question that WhatIf is optimized to answer. And it sort of underneath also does a comparative modeling for it. There's another method that I briefly want to mention. This COMA from Česlovas Venclovas. And the breakthrough or the advantage that COMA, and this I really have to update, but the advantage really is the way Česlovas is doing the alignment better than everybody else, and through this doing the alignment better, they improve over other methods. iTaser, Yang Chang is one of, again, a method that sort of came up in CASP because the fame of iTaser. This was the first. Somebody mentioned the name Baker here, David Baker. So this was the first thing that did beat, as an automated machine, David Baker's expert structures. And ultimately, what they do is something that looks similar to what the others do, but they do much more in terms of template query template matching. So find the right template, correctly assembling the fragments and correctly looking at spatial constraints that are really from predictions too. So here's one result from EVA was a server that checked, essentially did an automated CASP, a continuous automated CASP, and for years tested comparative modeling servers, queried comparative modeling servers with proteins that at the time of the submission were not known, and then when the structures came in, the server simply checked how well they did. What I show here is how well different modeling servers in terms of sequence identity do, and you see that throughout some range of sequence identity, these 5 different servers here, they are all below some 5 angstrom threshold. So if the sequences are similar enough, they all manage the task. And then, at some point, things get more complicated. So this is one.
How come they go down again?
Yeah, you see the error, right? I mean, maybe you don't see it because it's not very. So the error estimate. So they're huge error estimates because the numbers are too small there. So this was, again, so what is remarkable here, what I show you is something that I believe was like 40 or 50 CASPs. And after 40 or 50 CASPs, this was a continuously running server. It was running for 5 years. So over some period of these 5 years, you assemble many more than 100 proteins, right? I don't know, I believe this is 10,000 proteins. CASP is 100. 100 CASPs. So this is around 100 CASPs. And you see that after 100 CASPs, so after 200 years of CASP, you still would have immense error estimates. So you would still not be able to resolve. You essentially would see that the 4 methods are essentially similar here, and where they begin to fade apart, the error is so high that you cannot say anything. But there is something happening here. But the major point of this slide is something else. So this is accuracy. How well is your model? This one now is equivalent positional coverage. So this means essentially I have a sequence. How much of the sequence can I actually model? Because the sequence, that query that I start with, and the template, they are not 100% identical. We talked about the loop where they are different, but there are other regions where they're different. And what this one shows here is that even in the realm here of under 40%, you're still only modeling something like between 20 just a little bit more than, I don't know, 60 to 80% of the protein. So you're really not talking about the entire protein. And at some point here, you fall dramatically. And yes, you do get, when I said we get models for 40 million, but many of these models for 40 million, in fact, model something like 20% of the residues. Okay? Those are sometimes very short fragments. Is the homology model helpful? Well, better have a little bit of structure than none. That's ultimately the statement, but this is something that you have to be aware of. Yes?
Is there like a reverse method at play? Because this is all starting with a sequence and then looking at other sequences to find out possible structure.
Yes.
What if I were to say, well, I want to design an enzyme based on a reaction. So what I have as my template is actually electron density and maybe some dynamics.
Yes.
And then I would like to go into the PDB database and from that not extract the sequence, but I want the electron density and the dynamics that would fit my request. Let's put it that way. Is that
So essentially create all the sequences that fit to the structure.
Exactly.
Yes.
Exactly.
So there is, in fact, now we're back to David Baker. David Baker published, so he, as far as I remember, he was the first one who succeeded in, so several people have tried to do that. And it goes back to something that I suppressed here, but you may have seen fold recognition, you may have heard the word threading. Somehow underneath those are methods that I will not talk about because of brevity of shortness of time. But ultimately they sort of use that idea. And the first time that, as far as I remember, that this led to a moment where somebody actually put it into a tube and said this is the fittest and actually it adopts the three-dimensional structure was David Baker in the case. So several people have tried that exactly for design purposes.
Because let's say I want to have a function. I don't care about
design.
About the sequence.
This is exactly the design purpose.
And then shouldn't I have like more like wiggle space?
So again, my answer now was people have done that, but this is the kind of thing that does not work for every single case you try. So this is highly non-trivial, and we have publications where it worked.
Okay.
It does not mean that for most of the biochemical engineering of we talked about the iGEM briefly. So typically you move around in a much more constrained space. You change a residue, or you hope that you knock out the binding site, or you change the binding site, or but you create a completely new, or you find a completely new sequence that fits the structure. Again, there's a case where that worked, but this. For the next protein design task is to...
As the lady thinks, there's no big server like...
No, absolutely not, because it's too complicated. The search space is too big. Now, what I also want to, again, point out on this curve here, when you sort of run a PsyBlast search today, so you take a query sequence and you find whatever you can, the family of 1,000, and then you take these 1,000 and you ask, what is the pairwise sequence identity? Then you get a distribution like this red one. Meaning, essentially, when you run a PsyBlast and you pick everything below a 10⁻3, then the vast majority of things you find are down here. Which in fact means that many things that are in Swiss model of the 40 million are exactly here, where you have a coverage of very little. Right? So that's to be remembered when I say that we can, comparative modeling is the only method that completely predicts 3D structures at high level of accuracy, the only generic method, and it can do that for 40 million. Well, that's true, and that's a tremendous success, because the next method that can do that for maybe a few thousand, and we will talk about that maybe later, but nothing comes anywhere near that in terms of coverage. But really, most of it, again, this is a cumulative distribution, so in the cumulative distribution the numbers are missing, but sort of, if you estimate that this is 20% of the red distribution, right? So 20%, so of the million, only 1/5 of the 40 million, so 8 million, have coverage of more than 30% of the residues, roughly, right? Most of those have coverage less. That still, for long proteins, can still be long region, but that's the limit, okay? So it's nowhere near half of all the proteins have known structure. Half of all the proteins have some region, and typically a very short region, for which I sort of can infer a model. Okay? Having said that, let's get back into Cécile n'est pas une pipe. This is not a pipe, translated from René Magritte. What does it mean? So this comes together. This was done in 1929. So the image, the painting comes together with a statement. How do we interpret that? So this is not a pipe. What does it mean?